everyone, Learning Insight welcomes you to today's new topic, urine physiology. So let's have a look. Urine physiology. Here I have drawn a nephron out of trillions. So this nephron has Bowman's capsule, artery and vein as you know, and cortex and medulla. So here you have a particular concentration gradient called cortico-medullary gradient. There are 4 sites in formation of urine. Site 1 is at PCT, site 2 is at ascending loop of Henle, site 3 is at DCT, and site 4 at collecting duct. So let's see what has site 1. Here we have active transporters as well as passive transporters where sodium, potassium, and here the third one also sodium and hydrogen, all these are actively transported in a symport format, and water is just secreting out of the lumen into the cellular space, and chlorine and sodium, these are all passive transport as it is differentiated. Straight line is something called active transport and the dotted line is passive transport. So here when there is lot of concentration and now all the ions are getting removed, so now here it becomes isotonic to that of the concentration present in the medulla. So next comes the site 2 at thick ascending limb of loop of Henle. Here we have a symport, sodium, potassium, 2 chlorine, symport. Sodium is reabsorbed, potassium is reabsorbed, as well as Cl is reabsorbed. But one thing, potassium when it is reabsorbed excess, so potassium is regained back into the lumen because there is a basement membrane. When there is more amount of potassium reabsorbed, it gets back into the lumen again. So then we here because there is more positive charge. Now when potassium also came back, there is more positive charge in the lumen. Hence calcium and magnesium, they by passive transport, they just get out of the lumen. That is they are secreted out of the lumen. You have to remember one thing, that in thick ascending limb of loop of Henle, here there is no reabsorption of water, that is only ions are reabsorbed. Now, so what do you think, what will become the lumen, that is urine? So it will become dilute. Yes, there is more presence of water and less ions, it becomes dilute. So here there is hypotonic. Okay, so now we get to the site 3, which is DCT. Here we have sodium chlorine symport. Because of electrochemical gradient, chlorine is reabsorbed and sodium at the site 3, it is it is reabsorbed and again it comes back with the transport with the antipode of calcium, with calcium, and again there are so many transporters which help sodium to get reabsorbed into the lumen. Here at site 3, water is permeable to the membrane and hence water is secreting out of the lumen, and also chlorine as well as all these ions. Potassium again gets back into the lumen. So whatever the sodium which is left in the site 3, that again gets that is forwarded into the site 4. Here at site 4, till now there is decrease of ion and water is also decreased. So here from hypotonic it becomes into hypertonic again. At the site 4 we have aldosterone effects. Sodium enters the cells by electrochemical gradient, that is through amiloride sensitive renal epithelial channels. So but chlorine is not not reabsorbed to maintain that negative ion into the lumen, negative ion charge. So chlorine is maintained into the lumen. So sodium is reabsorbed, potassium is reabsorbed again, and it again gets back into the lumen. And as well here there we can see that there is an antipode of sodium and hydrogen, that is hydrogen ions, they get back from the ECF to that of the lumen. ADH is anti-diuretic hormone. So what do you think? Anti-diuresis, that is it is not allowing the urine to be dilute, so it is concentrating the urine, hence water should move out and the whole urine will be concentrated. So whatever now excreting out, out of 4 to 5 liters of blood, only 1 liter of urine is excreting out. Okay, so now let's see in complete description whatever I have told. Now we'll see it in a written format. Site 1, PCT. 4 mechanisms: direct sodium reabsorption, transport of sodium, potassium with glucose, phosphate ions, they are also reabsorbed. Next is exchange with H+, and also another one is bicarbonates and phosphates and acetate reabsorption. They create passive forces for diffusion of chlorine. So now let's see what is exchange with H+. How does it happen? Here a very important enzyme comes into place, that is carbonic anhydrase enzyme. And this enzyme is when you come to the treatment of for kidney, especially the diuresis, we have carbonic anhydrase inhibitors. So this particular mechanism is really important. So now let's see how exchange with H plus occurs. So we have seen that sodium and H plus exchange is there, right? So sodium, it gets reabsorbed, H plus gets from the tubular cell. Here sodium from lumen is reabsorbed and water is excreted out. So now how do you get water into the lumen? So we'll see there is sodium bicarbonate which is in the lumen, which is dividing into sodium and bicarbonate ions. Bicarbonate comes and this H plus is forming into H2CO3, and that is leading to formation of H2O and CO2. And now this H2O comes into the it is reabsorbing into the tubular cell, and here with the help of carbon dioxide is forming into H2CO3. Like whatever it was done here, the reverse thing happens here. H2CO3 then into HCO3 and H plus. Now H plus again, it becomes like the whole cycle again repeats. So here again there's HCO3 minus which is there, that HCO3 and Na plus which is getting reabsorbed, they become into NaHCO3. Now let's see what is site 2, thick ascending limb of loop of Henle. Here we have, as I mentioned, no reabsorption of water, only ions are exchanged, hence it becomes hypotonic. So sodium is reabsorbed, potassium is reabsorbed, and as such even the excess back to lumen and positive ions are increased, the same thing. Hence calcium, magnesium, they are again in those are removed out of the lumen. Now site 3, we have sodium and chlorine which are reabsorbed. Here sodium is sent to site 4, that is, as I said, forwarded. So now at site 4, which is collecting duct, sodium enters the peritubular cells by electrochemical gradient. Chlorine is not reabsorbed, as I said, to maintain the negative charge, and potassium is already present in the lumen, and here we have potassium is present in the lumen, so potassium is getting excreted out into the urine, right? So here we need in the treatment we have potassium sparing diuretics which comes into action here at this particular site. Okay, so this is what is about site 4. Here H plus is again coming into the in exchange with sodium by pumps in luminal membrane. Here the H plus enters into the urine, and as such it becomes slightly acidic. Now let's see what is antidiuretic hormone. Antidiuretic hormone is no diuresis, right? So this hormone inculcates no diuresis in the urine. So we'll see two aspects, presence and as well as absence. Absence, it leads to dilute urine, it leads to hypotonic fluid, and in presence it is leading to concentrated urine, and this is very important in conditions like swelling where the whole fluids should be removed out. Now aldosterone at site 4, as we talked about aldosterone effect over there, it will control the absorption of sodium, leads to efflux of potassium with sodium. So now one last thing, let's summarize. Site 1, sodium, chlorine, water, potassium, HCO3 reabsorbed. Site 2, sodium, potassium, 2Cl, calcium, magnesium reabsorbed. Site 3, sodium, chlorine, calcium, magnesium, water reabsorbed. And Site 4, sodium, water are reabsorbed. Hence let's come to the end of the YouTube video. Like, share and subscribe, and stay tuned.